members of the SBF. And it's a pleasure to have Florence Bansept with us today. She's the group leader of the Laboratoire de Chimie Bactérienne in Marseille. She's been a permanent researcher at the CNRS since 2024, and she's a fellow of the Turing Center. And it's a pleasure to have Florence Bansept. Oh, I'm hearing my own voice here. Of the Laboratoire de Chimie Bactérienne in Marseille. And she's a fellow of the Turing Center of Living Systems Centauri, MUI, and she's a permanent visitor at the Center of Theoretical Physics. She's been a postdoc at the Max Planck Institute for Evolutionary Biology at Plon. And she... I'm sorry, I'm hearing my own voice here. And she's a permanent visitor at the Center. I don't know what's happening, but anyway, she was here at URGS during her master back in 2014. And I think I don't need to make any new introductions. I'll pass the word to Florence. Hopefully she won't be hearing her own voice. I'm going to see what's happening here. Otherwise, it's going to be a bit difficult for you to talk.
Okay, let's try this. Okay, that looks good on my side. Good. So good morning, and thanks a lot for the invitation. It's really a pleasure to be talking here. And I'm going to tell you today a little bit about my work, or our work, on host-associated microbial communities, and with a particular focus on migration. So, as you may know, most multicellular organisms do not live in isolation, but they rather share a body with thousands, millions, billions of microorganisms that form ecosystems that are called microbiomes. So, for example, on a human adult body, you can find up to 10 to the 13 bacteria that are, which is roughly equivalent to the number of cells that form that body, and most of them are located in the intestine, where they perform some essential functions for the host, for example, helping it with digestion. There are many empirical evidence that show associations between a disbalance in the composition of the gut microbiome and some pathological states, such as obesity and diabetes. And for that reason, the prospect of one day being able to manipulate the gut microbiome for therapeutic purposes represents an important hope. But if one wants to one day be able to manipulate it, it's important to understand what rules govern the microbiome composition in a healthy host. And this is far from being a trivial question, because it's an open system where you always have incoming microbes, and you have interactions both between the microbial types in the community, but also with the host and with the environment. And you also have an outflow of microbes towards where. So the aim of my group is to use mathematical model as a way to decipher these different mechanisms, to understand their relative importance. And today I'm going to focus on two different topics around that general idea. So the first part of my talk will be dedicated to selection gradient and life history traits in host-associating microbes, which is a work that I have started during my postdoc, but that is still ongoing now. And in the second part, if I still have time, I will tell you about some more recent work on intermittent migration through feeding. So, fitness, when you talk about microbes in the lab, it's most often measured as a single replication rate in fixed environmental condition. But when you think at a larger scale, when you think about mammals, for example, it's, or not mammals in this case, because I have a figure with turtles, but bigger organisms. Each step of the life cycle can contribute to reproductive success, and that's a pretty simple idea. So, for example, for these turtles, in order to go from a new offspring to new offsprings. You need those baby turtles to survive up to adulthood. You need them to maybe migrate towards the appropriate breeding sites. And because this is sexual reproduction, you need them to find a mate to reproduce and produce new offspring. And microbes too, they can have some pretty interesting life cycles, in particular when they associate to hosts, because their life cycles can be very intertwined with the ones of their hosts. So for example, you have microbes that are going to use some larvae, some nematode hosts as vectors to travel from one insect host to the next. So a question arose when we were discussing those topics during my postdoc, and it was, how did microbes that a priori at the beginning of life on Earth were all free living because it was only microbes, how did some of them transition from this lifestyle to host association to a point in which today some of those associations are obligatory, meaning that the host cannot survive without the microbes cannot survive without the hosts. And the working hypothesis we've been using is that in this transition, biphasic life cycles have been key, but also biphasic life cycles can still be observed for many microbes today. And what we call, and what I will call in the rest of the presentation, biphasic life cycles are life cycles in which microbes spend part of their time living freely in the environment and part of their time associated to a host, and they are able to transition from one state to the other. And the question I wanted to answer as a modeler were, how can we characterize the life history traits contributions to fitness? Is there an optimal strategy for the microbial population to maximize its fitness, and how can we measure this? And in order to answer those questions, we developed few minimal models to try to quantify these things. And all of these models, they are models of biphasic life cycles. So the first model that I'm going to talk about is a more or less epidemiology inspired. It's an SI type model where you have 4 different types of compartments and you follow the dynamics of the compartments and not of the microbes themselves. So in this model, you have 2 types of hosts, either free of microbe, which we call susceptible for the analogy with the SI model, or colonized by microbes, which we call I for infected. And the same for environments. You can have either susceptible or infected environments. And the susceptible environment, they can become infected when they are entered by infected hosts. And in a symmetric way, you have susceptible hosts that can become infected when they enter into an infected environment. You also have some fresh environment influx, which you can think of as apples falling from a tree, for example. And you have some logistic growth of the hosts with no vertical transmission, meaning that the term of host growth that comes from the infected hosts still appears in the non-infected population. And on top of all of that, we added a little bit of death for all of these compartments, death, decay, something to make them not explode. And with this very simple, very minimal model, what you can do is simply look at the proportions of infected habitats and look at this in function of the products of the transmissibilities, lambda and omega, which were the rates by which susceptibles became infected. And what you see is there is some sort of critical effect, some sort of R0-like criterion. So below a certain value of the transmission product, there is no propagation, and above it, there is a finite proportion of hosts and environments that become infected. And you see that this critical effect depends on the product of those two transmissibilities, which suggests that there is some trade-off between the two transmission routes. And this is how we understood it. But this is a very simple model that does not distinguish between the habitats that may contain thousands, billions of microbes and those that may contain just a few, because we do not keep track of the microbial populations themselves. So what happens when we do keep track of the microbial population? That's what we looked at in the next model, which is just as minimal as the previous one, if not even more, in which we looked at a microbial population that is shared between a host compartment and an environmental compartment, which this time are fixed, so there is no dynamics of the compartments themselves. But within each of the compartments, the microbial population can replicate at maybe different rates, RH and RE, and there is some migration between the compartments. And from this, you can write the population dynamics as a matrix population model where everything is linear because there is only exponential growth, no competition of any sort in the base model. And you can look at the largest eigenvalue and you can look at how the total population grows at long times. And that will tell us something about the reproductive success of the population that is characterized by certain rates. And what you can see on this plot is that there are two strategies to increase this global population growth rate. Either increase the within host replication, so that comes from the assumption that initially the microbial population is well adapted to the environment and replicates slower within the host. So this is quite intuitive. But the second strategy is to decrease migration. Initially, we consider that both migration rates are equal. And this just comes from the fact that everything is exponential in this model. So what matters the most is that once you are in the compartment where you replicate well, you should stay there. And it doesn't matter if you sacrifice the population that stays within the host. But if one wants to answer the question that follows, which is if I'm a microbe that starts from a specific point in this phenotypic space and I want to increase my fitness, but I can invest only in one jump in this phenotypic space, so should I first jump towards the right or jump towards the bottom, to say it very simply? And this you answer with doing some sensitivity analysis. So just look at the gradient of this quantity with the different dimensions of this space. And what you find is this separatrix that shows you that above the line, the strategy that is the most efficient is to increase replication, and below the line, it's to decrease migration. So it's a very simple result, and yet it really emphasizes that sometimes migration is the key factor in increasing the fitness of a population, and it's not just about replication rates. And that is going to depend on where you start from in this phenotypic space. So then we did some refinements of this base model by looking at initially asymmetric migration rates. And you see that it doesn't change much the general behavior of the optimal strategies, except for the slope of that separatrix line. And we also added different kinds of competition. Here I showed just when there is within host competition. In this case, the system becomes nonlinear and you need to resort to actually looking at population sizes at equilibrium. And when you vary the strength of within host competition, you will slowly get towards the equilibrium, which you calculate from the equilibrium point of your system at long times, which comes from that non-linearity. And the intermediate lines, they actually look at a defined time in space, which was actually set from experiments, but I will come back to that right after. So the conclusion holds that depending on the starting point in the trait space, the selection gradient can point towards different optimal strategies. So yeah, I just hinted at that. Those minimal models, they actually emerged from discussions with experimentalists, and they were performing super interesting experiments, which I'm going to present now. So those experimentalists were mainly Nancy Obeng, who at the time was a PhD student and then postdoc in the group of Heinrich Schulenburg at Kiel University, Germany. So in those experiments, they took some bacteria that they found in the gut of some wild-type C. elegans, which they collected in the compost of their university. And they put those bacteria in different conditions. So either they put them on a petri dish for 3.5 days. They let the bacteria freely develop on the petri dish, and then they added some worms that were free of microbes, that had been bleached experimentally to be microbe-free. They let the worms feed on the bacteria for 3.5 days. And after that time, they retrieved the worms from the experiments. They cracked them open and retrieved the bacteria that were inside the worms and get a fraction of them to start the new evolution cycle of this experiment, in which, as you understood, they actually impose some biphasic life cycle where the bacteria live part of the time on the petri dish and part of their time in the worm. And they compared how those bacteria evolved compared to ones that had been put only on petri dishes, in which the evolution experiment is also repeated. So after each cycle, you take a fraction of them and you put them on a fresh petri dish, but in which there is no worm. And in order to characterize how differently those two lines evolved, they did some additional phenotyping of those bacteria. And they looked, for example, at their ability to colonize worms, to stay in the worm, or to exit the worm. Also, their ability to move on the petri dish and also to grow on the petri dish itself. And so you see that in those experiments, what is actually measured in those follow-up phenotyping experiments are sizes of bacterial population. So if you think about the two toy models that I presented, it's actually like the aspect of the second model, which is actually keeping track of the microbial population itself, is important. But also in just one single cycle of these evolution experiments, the number of worms is going from initially 10 at this point in the experiment to several thousands at this point because they reproduce. So the aspect of the first model I talked about, where you have some dynamics of the habitats themselves, also matters. So if we want to be able to predict anything about what happens to those bacteria, or at least to quantify the selection gradient that applies to them, it was necessary to combine those two types of models. And this is what we did here, trying to keep the simplest possible version, in which we just follow the size of the population of microbes that is associated to the gut, that is living inside the gut. And that's because we always assume that the population of microbes, of bacteria, is in excess on the plate, which is something that was observed experimentally. So this population, it increases when the worms are feeding on the bacteria that live on the plate. And you see that this is limited only by the worm compartment, which is called W here, because bacteria are in excess on the plate. And they increase also with logistic growth within the host that is limited by a carrying capacity, which you see depends on time, because it also grows with the development of the worms from larvae to adult stage. It decreases with bacteria either being digested by the worm, because this is what they use as food, or simply being expelled alive on the plate, which is also something that is observed in the experiments. And yeah, as I said, this the worm compartment is increasing with time, with the worm's development, and we took this as being linear because it's more about worm development than actually looking at several generations of worms. And this time we simply track the population size and look how it looks like after 3.5 days, which is the experimental time during which the bacteria and the worms were living to interact with. And what we find is summarized on this slide. So this is the result of the sensitivity analysis that has been performed on all the parametric space. And in this case, the parametric space is just constrained by biological estimates on all those parameters of the model. And what we find is that increasing the replication rate within the host is actually almost never optimal across the whole parametric space. And the strategy that is most often optimal is to decrease the rate at which the bacteria gets either digested by the worm or expelled alive on the plate. Then, of course, this is just an aggregate of the whole parametric space. So it's also interesting to see how that segregates in the different directions. And this is what you can see here. So yeah, these two dimensions, they separate rather well the three most often optimal strategies. And then the hope was that we would be able to have several experiments with several types of bacteria that would maybe live, at least initially, in different parts of this parametric space so that we could compare their evolution. But unfortunately, it was a lot of experimental work. And it was also extremely difficult to do any inference of those parameters from the experiments. So what we have is only some qualitative comparison, but I'm going to tell you the results of the experiments themselves. So first of all, something happens which cannot be accounted for by our models, which consider fully homogeneous populations of microbes. And this is the fact that different morphotypes emerged. So in particular, in the biphasic line of the experiments, there were some wrinkly types that appeared and that have been observed in other types of experiments in completely different contexts. But because they looked like this, the experimentators we were working with sort of knew in which direction to look at. And it was confirmed by the phenotyping experiments they performed because what they could measure is that those wrinkly types that are most often associated with the host, they get better at forming biofilms. And this is also something that they could check with simply looking at the genotyping of these different types. And this, we want to argue, is compatible with the strategy of decreasing the environmental release towards the environment. But of course, our model is very limited in terms of what can be predicted. And in particular, as I already said, regarding the differentiation into different microbial types. So the last thing I'm going to tell you about this work is some work that is currently in progress in order to develop a model of biphasic life cycle in which differentiation is actually allowed. And this project we are developing in collaboration with Cécile Carrère, who is maître de conférence at the University of Orléans. And it's actually a sort of a random thing that happened, that she heard me talk about everything you've heard so far. And she said, oh, but that really reminds me of a whole body of literature in applied mathematics. And now we are really trying to connect those things. And the way we're doing that is developing a model that is phenotypically structured. and in which we add one dimension. So we get some PDEs in which the first dimension here, X, is going to be, I'm going to call it the genotype, even though in the literature, most often it's called the phenotype. So the NE now states the density of bacteria that have a certain genotype at time t. And here the number of bacteria that live within the host also at time t. And you see that those can diffuse, which is going to account for the mutations around the genotype of the mother cells. But also this genotype translates as a different phenotype, so different replication rates in each of those two compartments. And actually those functions of replication rates, they look like this, with an optimum that is going to be different in the two different compartments. So if you replicate within the worms, within the host, maybe it's going to be optimal that you have a genotype that is around -1, and when you replicate within the environment, it's going to be optimal if you replicate with a genotype at 1. And so there is logistic growth in each of those compartments with those replication rates that translates this genotype-phenotype correspondence. And there is also some migration between the compartments, and the migration from the environment towards the host could depend in principle on the host biomass to reflect this aspect that the hosts feed on the bacteria, so the amount that is actually ingested depends on how many worms you have on the plate. And I'm not going to show you results about that part because we are still thinking about how to tell that story. We have generated a lot of data, and in particular, now there is Leon Espinosa, who is a research engineer in my group, who's been developing an agent-based version of this model. So Cecile is working on the PDE aspect, and Leon is working with me on the agent-based version. And the aim is to have a good representation on in which case we are expecting to see differentiation in the microbial population or not, whether it's going to be better for the population to keep some sort of generalist genotype that will allow them to transition quite easily from one habitat to the other without ever being at the optimum. And that, for example, we expect to be the case if the two optima are not too far from one another, or in which case it's going to be better to really specialize in the habitat in which you are living, even though that means that when you migrate, you're going to have a really hard time. So we are building this kind of maps, and hopefully another time I can tell you more about this. So that's for this first part of my talk. So I told you about several simple models of biphasic life cycles in which microbes can live part of their time in the environment and part of their time associated to hosts. I showed you a first model that was inspired by epidemiology, a second one in which we keep track of the microbial population itself. I showed you what happened in the experiments and what we were able to say when we take aspects of both those first models together, and how we are currently developing those ideas to take into account the heterogeneous aspects of the population. But I think if you have to remember just one aspect of this talk is just that migration actually matters when you talk about microbial fitness, when you think about the reproductive success of a microbial population. And in the second part of my talk, I'm actually not going to go too far from that, because I'm going to tell you about, again, a story of migration. And this time it's going to be the story of immigrating microbes through feeding. So first of all, this work has been recently submitted and is available online on bioRxiv. It's the work mostly of my postdoc, Vitor Marchioni Monteiro, who's also Brazilian, and of two previous master students, Anne-Catherine Hoffacker and Jackson Villavicencio. Okay, so just a little introduction to that topic itself. So I told you in my introduction half an hour ago that we wanted to understand the rules that govern the gut microbiome composition in a healthy host. But what does the gut microbiome look like in a healthy host? Well, some experiments, well, many experiments actually, show that there is a link between the diversity of the gut microbiome and health, but that link is not always that clear, in particular because it looks like lower diversity states are indeed associated to pathological states, but it's not clearly the case that if you keep increasing the gut microbiome diversity, you're just going to keep bettering your health. From a theoretical perspective, some links have been established between the diversity of a community and two properties which you would like to see in a healthy host, which are resilience and stability. So essentially, the ability to go back to your initial states after perturbation, which, if you are in a healthy state, you would like to have. If you're not convinced by those two previous experiments, maybe we can try this thought experiment of thinking about the extreme case of pathogenic infection. So in this case, you have a pathogenic agent that enters into the gut microbiome and opportunistically maybe takes over the whole community and becomes a really substantiate proportion of this community. And in this case, of course, the diversity of the community drops. Maybe you're not convinced by any of this, but I'm going to use my last argument here, which is the practical one, which is that it's extremely complicated to really characterize microbiomes of healthy hosts, and using diversity is a way to reduce this complexity to just one number. So in the rest of the talk, I'm going to shamelessly use the microbiome diversity as a proxy for host health. And hopefully you don't find this too shocking, but even if so, I think even if diversity does not equal health immediately, I think it's still interesting to look at the drivers of diversity in the gut microbiome. So it looks like it's in the interests of the host to somewhat control what is in its gut microbiome. And maybe the first thing you're thinking about is that the host could just use its immune system, right? But actually, in the gut, it's going to be quite costly and difficult to use the immune system. First of all, because any effector that you produce within the gut is going to be evacuated ultimately towards you know where. And also because different microbes that live in the gut may look very similar, but have very different effects on the hosts. Some of them may be perfectly commensal in normal conditions, but then you have a perturbation and they become opportunistic pathogens. You can also have evolution within the host. And for all these reasons, it's really difficult for the immune system to decide, make decisions on who can stay and who should go. So we are more interested in solutions that do not require to make this decision. And for example, the host can actually control what is coming in. And this is an idea that has been quite studied, but more from the perspective of the nutritional content of food. But we wanted to start with something that we found as physicists to be more simple, which is to look at the microbial content of food. Because it is shown that some of the microbes that you ingest are going to establish in the gut community. That's the whole concept of using probiotics, for example. And that's true even though in most animals, there are some mechanical, physical, chemical barriers that are going to kill a lot of the microbes that you ingest. We thought that it was interesting to focus on that, also because in theoretical ecology, it is known that immigration is a key factor to the maintenance of diversity. And also because there are some temporal patterns to this migration through feeding. In most animals, they don't feed in a continuous way. There are some periods of time during which the community within the intestine is almost isolated from the environment. And those temporal patterns in theoretical ecology, in models in which you have birth, death, and migration, they most often have been neglected. So we wanted to ask the following question: is there an optimal feeding frequency, an optimal feeding strategy for the host to optimize its diversity? If yes, does it depend on host-related parameters such as typical transit time, carrying capacity, etc.? Because we do not expect this effect to be as important in humans that wash their food, that have quite slow transition times, and in which it seems that colonization in early life is what matters the most. We expect this situation to be very different from, for example, C. elegans, in which the transit time is about 30 seconds and in which the host actually directly feeds on microbes. The additional question is, when should experimentalists control for sampling time with respect to feeding? So does it matter whether we take a sample of the gut microbiome right after having been fed or a couple hours later, a couple days later? And in order to study those questions, we built a minimal model in which there is an infinite pool of microbes with fixed proportions of the different types. And from this infinite pool, there is a flux of microbes with a certain temporal pattern that goes towards a local community within a host, in which microbes replicate at different rates, but also get cleared towards still, where, and where there is also a carrying capacity that makes the different types interact with each other. And I'm gonna build that model really slowly. So I'm sorry, I'm gonna try not to be too slow because I think I don't have that much time anymore. But when you have just one species, this is how the equation looks like in absence of feeding. So you have some logistic growth of your microbial type, you have some clearance, and of course, how the abundance looks like in function of time. In this case, you saturate at a value that is slightly below the carrying capacity which you put in the model because of clearance. And if you add continuous migration on top of that, you are going to saturate just a little bit above towards a value which you can also calculate analytically. If you make now this migration something intermittent with bursts of migration of size NF every tau, and you keep NF over tau a constant, so that's the integral of the food amount that reaches the gut per unit of time remains the same. You see that you have those bursts that get you above the carrying capacity, and then some decreases in a cyclic way. And if you increase the feeding frequency, well, the fluctuations become smaller, and of course you can see the frequency of the fluctuations that also increases. So that's not very surprising. Now let's talk about the model when you have two different species, and in particular when you have species 1 that is not doing very well inside the environmental pool, that is present in very small proportions. And in the host, on the contrary, you have the blue species that is doing really well and that is replicating faster than type 2. So in this case, if feeding events are too rare, what is going to happen is that inside the host, you're going to have the elimination of the orange type because of the shared carrying capacity. There is going to be competition, and the blue type is going to eliminate the orange one. Now, if the feeding events are too frequent, there is the risk of losing type 1 inside the host because the community inside the host is going to resemble the composition of the pool of microbes. And if you add some stochasticity on top of that, then you might lose type 1. So with this very simple concept and this very simple anti-correlation between those two quantities, those two things, you are in a situation where an intermediate frequency should optimize within host microbial diversity. And this is the whole idea we started with. And this you can indeed check. So this is the case where you don't have migration and you then lose the microbial types that has the largest C over R, so clearance over replication rate. But if you add some immigration, you can recover the type that you were losing before. Now I'm actually going to switch towards using diversity index because it's all simple when you still have only two types, but it's going to be slightly more complex. So in theoretical ecology, diversity is something that measures both the richness of the community, which means how many types in your community you have, but also the evenness. And the evenness corresponds to the following thing. It corresponds to how even the different proportions of the different types are in your community. So in the first community here, the three different types, so both community 1 and community 2 have the same richness because they have three different types of microbes. But in the first case, the three different types of microbes, they are represented in equal proportions. And in the second type, one of them is way more frequent than the others. So that's the whole concept of measuring diversity, and you have different ways to combine those two things. It could be the variance, the Gini index, the Shannon index. In the rest of this talk, it's going to be mostly the Shannon index, but also a little bit of the Gini index because I did not redo my slides when we switched. Sorry about that. And when we look at this diversity index in function of the feeding parameters, which are the size of the bursts of migration and the feeding interval, what you can see is that indeed there are some feeding intervals that maximize this diversity, that the effect of actually the slope, so the ratio between NF and tau, is what seems to be the most important to determine the diversity of the community. And that the position of this maximum actually depends on the proportions of the types in the environmental pool. Also, you see that the constraint on tau relaxes when you get larger microbial influxes. So that's fine. It actually works exactly as we imagined. So we actually built a model for our initial idea to work, and it works. That's not very impressive. So what was actually interesting was what was going to happen when we add more species to the mix. And this is what happens when you have 3 and 20 species, and in which, most importantly, there is no correlation between the replication rates and the proportions within the environmental pool. And you see that there still does exist an optimal feeding strategy. So it's at this point of my explorations that Vitor joined, and all his work was about developing some approximations to fully characterize the system and some numerical analysis too, to see in which cases this optimal feeding strategy was going to exist and in which not, and to see whether we could understand how that works with large numbers of species. So I think probably I don't have the time to tell you everything about it, but I'm going to try my best before I get stopped. So between feeding events, we considered this equation for type I of microbes, so still with logistic growth and some clearance. If you want to integrate that equation, you see that this depends on the total abundance of microbes if you sum all the types. So what we did first was just have a trapezoidal approximation for both the total amount of microbes. So just approximate this integral as this trapeze below the curve that is defined between t0 and a t0 plus tau. And also do the same thing for each of the microbial abundances, which allows you to solve that equation that then gives you the NLI, which are the base levels between which each of the abundances are oscillating. And this you can then inject into our diversity index, the Shannon index, and we can look how good it approximates the numerical resolution of the full system. And you can see that this approximation is good when the time between feeding events is low enough, and more specifically when this is validated. Another approximation which you can look at is the continuous feeding limit, similarly to what I showed in the case when you have just one microbial type. And that also allows you to look at the fixed point of the system, which still depends on the total abundance. And that allows you to look at, first of all, what happens with two species when diversity is maximal if the two species are present in equal abundance. And that allows you to get the optimal feeding rate. And then you have this line, which is the optimal, and which you can trace on the parametric space, which I showed you before, where you have tau, the time between feeding events, and NF, the size of the immigration births. And the optimal feeding strategy, which is characterized by the line on which diversity is maximal, is here the white line, and is really well approximated by this linear approximation. And it's a little bit above, so it's a supralinear regime. And Vitor likes to talk about it by saying that if usually you're eating one slice of pizza every tau, but you want to eat less regularly, and you want to eat maybe every 2 tau, if you want to have a diversity that is equivalent, then you need to add a little bit extra, so maybe something else than pizza, otherwise that would be too much. A very important result that we showed is that the existence of a feasible linear approximation to the optimal feeding strategy is a sufficient condition for the existence of a feasible optimal feeding strategy. So that means, and this is true not only for the case where you have only 2 types of microbes, that if we can solve the equations, so this equation in the case with 2 types, which corresponds to something we got from the continuous limit approximation, it means that even if you solve the full system with the temporal patterns and the intermittent feeding, there is an optimal feeding strategy. So I'm going to pass real fast on what is next. So when you have more types, you need to resort to Lagrange multiplier because there are too many constraints. You cannot just say that all microbes are present in equal proportions to maximize diversity. And in order to look at when it's possible to solve those equations numerically or not, what we did were some numerical explorations in which we randomly draw parameters from distributions, so both for the replication rates and for the clearance rates and for the proportions within the environmental feeding pool. And what we show very quickly is that when you increase the number of microbial types, there almost always exists an optimal feeding strategy. And we can actually fully characterize it in the case of many types. So we can show that the optimal feeding rate converges towards the average of the clearance rates times the total abundance of microbes, which converges towards the carrying capacity of the model. So I'm going to pass on that. I'm just going to say that it looks like what we find does not really apply well for humans, which is not at all a surprise, because there are many simplifying assumptions in our model that we know are especially bad for humans. So I would say that the exciting perspective for us is to get this tested for different animal hosts, because for humans, it's possible to find some orders of magnitude estimates in the literature, but it's impossible to find any for other animal types. But we are talking with experimentalists, and some of them are really interested in those ideas. So we've been talking with Tobias Lenz and Anna Teles from the University of Hamburg, who work with stickleback fish, and they have performed experiments in which they feed their fish either every day or every 2 days or every 3 days, and then they look at whether there is an effect of the sampling time on the microbiome, the gut microbiome. So either they sample on the day they're fed or the day after or the day after. And their preliminary data shows that it does make a difference. So that's very interesting. And we're talking even more with Thomas Röder and Anne-Catherine Hoffacker, who was a master's student with us and is now a PhD student with Thomas. And they work with Drosophila melanogaster, and they've done some preliminary experiments in which they focus on feeding times and the effect on lifespan, because in Drosophila, microbiome has a shown effect on the lifespan of the host, and we really hope that we get some more data from Drosophila really soon. So yeah, I think I'm gonna stop there with this idea that migration is important for microbial fitness and also for diversity in microbial communities. And I will stop with a thank you to all of you who were there, and most importantly, thank you to all the people who participated in this work one way or another. People from my postdoc on the left, and Victor and Anne-Catherine and Jackson for the second part.
Thank you very much, Florence, for your very nice talk, which covered both modeling aspects and experimental aspects. So now we will open the questions up for anybody who has questions on YouTube, and also for Francisco if you have any questions. First, I'll begin by a question that was already posted during the talk. So regarding the intestine flora, are there any studies related to the decrease in bacterial counts as a result of ultra-processed foods?
In the sense that there would be more total abundance of microbes as a result of eating processed food?
Yeah.
Okay. I'm not aware of the effect on the total abundance, but I know of many people who work on processed food for sure. And I do know that it's associated with health effects for sure, and effects on diversity, but effects on total abundance, I'm not aware.
Okay.
But
So
if so, I would be super interested in getting a reference about that.
yeah. So now we don't have any other questions, but I have a few. In the beginning, in the one of the minimal models where you showed the slides, I was a bit confused. You showed that you could change to the optimal strategy by going two different ways. Is that in the sense of bistability? Could you show us a slide, please, in
Yeah.
the...
So no, it's not in the sense of bistability. It's way more simple than that. It's just in the sense that the gradient is stronger towards the right if you are above this line than it is towards the bottom. It's as simple as that.
Okay. Okay. So, and then something else that I didn't, in the part where you work with the C. elegans,
Mm-hmm.
you show that you have an output from the worms with a new type
Mm-hmm.
of a phenotype. So I was kind of thinking because it's a system where I guess the bacteria don't want to be digested, they want to live, and while the C. elegans wants to have a nice meal. So there's a competition of these two factors. And then you show that the I think the wrinkly phenotype has easier biofilm production, if I'm not...
Yes. Mm-hmm.
Yep. So I think my question is that you also showed in some slide that having a lower time of output is better also. So I don't know, I'm not a biologist, but I would guess that by producing a biofilm, they would be a little more difficult to excrete. But at the same time, I guess the biofilm protects them from digestion. So could you comment on these
Yeah,
things?
that's a super interesting question. So I think maybe the experiment is a little bit biased in the sense that with those experimentally imposed biphasic life cycle, in order to get towards the next cycle, you actually need to have been inside the host. Because those 10%, they are not 10% of the whole of microbes that you found there. They are really the 10% which you could find associated to the hosts.
Okay.
So you're actually selecting for bacteria that are able to associate with the host. So in
Okay.
this sense, I think it's a little bit biased and does not reproduce correctly what happens in nature. Another aspect is that the worms that you put there in the experiments, they are always fresh worms in the sense that they are clones, so they are not able to evolve with the bacteria. So you're not measuring coevolution in this experiment, you're just measuring the evolution of the bacteria. I,
Okay.
I don't know if that fully answers your questions or not.
Yeah, I don't know. I think I'll have to read the paper because I found it very interesting because you have these two aspects, one from the worm's point of view and one from the bacteria. And also a question which is from a physicist, not a biologist. I imagine that there's a different carrying capacity for each type of bacteria, but how big would be that difference? And even if it's quite large, would that make any difference in the last part of your talk?
In the
Because I
last
think
part. Yeah. Yeah, that's also a good question. So in this last part, we are assuming a shared carrying capacity. So the model does not allow for different carrying capacity for each type. Otherwise, we would get rid of any type of interaction between the types. So in a way, having this shared carrying capacity, it's a way for us to take into account some sorts of interaction in a very simple way. But of course, it's a huge assumption. So the plan is to go towards full Lotka-Volterra description, generalized Lotka-Volterra. We have an intern who will start in that in January. So right now it's a little bit hard for me to answer that because we don't know yet. But what is certain is that in this model, what is very limiting is that you have this sort of mass effect that makes it that in absence of migration, there is only one type that can survive. And this is biologically really far from what happens in humans, I think. So I think in humans, you can have bacteria, even if you start eating only sterilized food, I think you can have bacteria that manage to maintain themselves in the gut. But for different types of hosts, like Drosophila, it is shown that some microbes can be lost over time if they are not present in the food anymore. So maybe it's a model that is better suited towards this type of organism. That's the only thing I can say so far, but it's definitely something we're interested in.
Okay, thank you very much. Francisco, do you have any questions?
Yes. First of all, I'd like to congratulate for your excellent talk and very interesting work. And as a panelist say, I will also read the paper you just make here, valuable. I think that I'm at some level repeating the question of handling, but I'd like to ask if you test to check if how many solutions you have for the stationary state, because you show a lot of mechanisms that are related to bi-stability, or even more a higher number of stationary states, like the competition, positive feedback, and the because this kind of ordering has been done for a long time, but without, but you have a lot of data. So my question is, have you checked the possibility of different number of stationary solutions for the stationary states?
So what I should say is that so in our model, in the full version of the model, because of intermittent feeding, you don't have an equilibrium. You have some periodic solutions. And the way that we look at diversity is getting some temporal averages. And actually, in the paper, we have a section that looks at different ways of averaging to see whether that has any impact on our results or not. And we show that it has only minimal impact. And that was important for us because the way that we average, which is the most simple way for us, is slightly different from the way that experimentalists would average their data. So that's the first thing. Another thing, so yes, maybe what I did not say about going towards generalized blood-cavity ultra. So I started talking about this mass effect, but I didn't say the second part of this reasoning, which is that if you go towards generalized blood-cavity ultra, then it's possible that even without migration, you have several possible steady states. And maybe that migration is going to allow the system to go from one stationary state to some oscillatory solutions around another stationary state. And that is interesting because maybe it's going to be possible to relate those different stationary states with different health status of the host. And maybe a third thing I wanted to say on the topic is, again, the influence of the carrying capacity. So when we go towards large numbers of, large number of microbial types, actually the carrying capacity does not have an effect anymore, in the sense that you do have the total abundance that goes towards the carrying capacity, but you get the same types of results exactly, whether you take the carrying capacity being equal to 1 or to more. So we renormalized all our numerical simulations because it was faster that way. I don't know if that answers all of your questions or not, but
Yes, absolutely. It goes beyond. Thank you very much.
thank you.
Florence. So I'd like to thank you once again for your time and for giving us this very nice talk. Since there are no more questions on YouTube, I think we can end the transmission now. Good
Wonderful.
morning, everybody. Thank you. And good afternoon to anybody in Europe.
Bye-bye. Thank you.
Bye.
Bye.